right, we are live. Welcome everybody to Standing for Truth. My name is Donnie, and I am your host and moderator for tonight's debate between Jamie from Studio 215 official YouTube channel and Taylor from the Snake Was Right YouTube channel. This is going to be a technical debate. This topic, specifically single to multi-cell evolution, and the scientific papers associated with it have been discussed in debates over the last several years, but especially over the last year in our 2022 Evolution Debate Challenge series. But never has there been an entire debate devoted to this important topic. So this one's been set now for a month or two. There's a lot of hype for this debate. I've had a lot of messages come in over the last month asking me, hey, when's that debate between Jamie and Taylor? So obviously this is a topic that people are interested in seeing engaged. And not only for a few minutes, within a greater debate on evolution, but an entire 2-hour debate on this. So we've got Taylor and Jamie, who have both done a ton of study on this topic. And that's why we're here for this epic technical debate on Chlamydomonas, single cell to multi-cell ? With basically Snake taking the affirmative tonight. So what I like to do, though, before we get into the fun, before we get into the debate itself, is break the ice, get to know our debaters a little bit. And Jamie, since this is your first time here, I do appreciate you being willing to engage in these important debates. Why don't we start with you? A little bit about yourself, how you doing today, and a little bit about your channel.
Yeah, so my name is Jamie. I have the Studio 215 official channel. The whole reason I started it was because I was raised as a Christian, and then as I grew up in middle school, they started pushing evolution aggressively. I fell for the trick, and then after I fell for that trick, it wasn't until my son was born, 15 years later, that I started to study the whole process of embryology. And that's what kind of sparked my passion for science. And then that's what led me down the rabbit hole that eventually showed me that evolution is a fraud. So that's what I do with my channel. I basically take evolutionary concepts and break them down.
Jamie, thank you so much. Chris Peacock, I appreciate it. I believe I had my laptop mic on since I refreshed everything today. Does my audio sound different and improved now, Jamie and Taylor?
Yeah, sounds better.
Sounds fine to me.
Okay. Sounds okay. So I'll take that as improved. And Chris, if you could let me know in the chat as well. So Jamie, I appreciate it. Your channel link is in the description box for people to check out. And Taylor, why don't we hand it over to you for a brief intro? You've been here before. You're no stranger, of course. A little bit about yourself and a little bit about your channel.
Yeah, so as far as this debate goes, this is pretty much my wheelhouse because I majored in molecular, cellular, and developmental biology. And so that was part of the original intent behind my channel, which is I wanted to kind of look skeptically at a lot of different topics, including, theology, politics and stuff. But I wanted it to be scientifically informed, and especially, like source methods process, because there's a lot of stuff out there that people claim without actually going to the sources. I also want to kind of challenge my own side, which is like the anti-theist side, because I think that they're infested by moral relativism and socialism, and I want to challenge those things. And one of the ways that I like to that I plan on taking the channel, one of the directions is, since I haven't had a lot of time lately, but gearing up towards that direction, is basically exploring fiction and the power of and meaning and spirituality behind mythology, and how that's kind of the soul of our civilization. And so I have kind of an atheist spirituality type angle. So trying to do some different stuff to the usual, just normal creationist theist debunkings. And I run a debate channel with my best friend. and she has a channel called Cinematic Psychology. Our channel together is Debate Cafe, which some of you might know. And, you know, anyone is welcome to come debate over there. Where it's jamming right now.
Awesome. Thank you, Taylor, for that intro. Thanks for the intros from the both of you. And Snake, I've got your channels listed, or at least I should, in the description box as well to Debate Cafe, where, yes, you've had some very solid debates there on that channel. You know that I always appreciate a good debate. So with that being said, for the audience sake, I will go over the format for tonight. Again, the debate tonight's a technical debate. We are focusing on single to multi-cell evolution and focusing on several of the technical papers in the scientific literature that we've discussed over the last year or so. But tonight we're really going to dig deep. So I'm pumped for this. So format wise, 12-minute opening statements. We're going to be starting with Snake with his opening. Then we're going to have 8-minute uninterrupted rebuttals, followed by everybody's favorite part of a debate, a 40 to 50-minute discussion. So rather than a really strict cross exam, we're going to allow it to carry on more free-flowing and organic, where Jamie and Snake here are asking each other the relevant questions to the topic of the debate. And then we'll have a 5-minute closing statement where the debaters can wrap up their thoughts and points. And then we are going to get you guys in the audience involved. We're going to have an audience Q&A about 25 minutes. So please make sure you're tagging me with your questions. Tag me at Standing for Truth. And then that way it'll be easy for me to spot your questions in the live chat and I'll save them. So with that, Taylor, we're going to hand it over to you. And so whenever you're ready, we're going to give you the floor for 12 minutes. Go ahead. Oh, I think you might be on mute.
Yep.
Oh, okay.
If I share my screen, can you just make me large? I mean,
sure.
Not when I share my screen, not if.
Oh, yeah.
All right.
There we go. Yeah. You're good to go.
All righty. So today I'm going to be arguing that we actually have observed the evolution from unicellularity to multicellularity in real time in the lab, thus proving macroevolution and the evolution of new information. And Jamie, I saw your original video when you tried to debunk my arguments on algal multicellularity, evolution experiments made during my debate with Kent Hovind. Mr. Hovind responded that he knew nothing about these experiments, despite the fact that I told him about them, quoted from them, given him the names of the papers over and over and over during our debates. He said to my face that he'll look into it each time, and each time he came back claiming no knowledge about it. So I castigated him a little bit for doing no research and repeating other arguments that he'd previously conceded in our other debates that were scientifically false. For some reason, he never uploaded this debate to his channel. I've heard that he only does this when he thinks he's lost. But that aside, he did, however, have you on his channel like not even 2 days later, where you gave your take and then challenged me to either give up evolution and convert to your religion, which I don't see myself doing anytime soon, or to debate you on the topic. So I chose the latter. So that's kind of the backstory behind this debate. And then quick point, even if I'm wrong here, that doesn't disprove evolution, nor does it prove any religion. That would be the God of the Gaps fallacy. So I would not be able to do that with any reason. Anyway, I regret to inform everyone that I don't think this video is available any longer. Correct me if I'm wrong, and send me the link, because I looked at Kent's channel and it's no longer there. According to my search attempts, I couldn't find it on your channel. So I mean, I'd love to see a re-upload or something. But yeah. So on to the presentation proper. All right, so the topic at hand, I was told the resolution would be these algal experiments in question demonstrate the plausibility of multicellular evolution. So I'm here to argue that not only do they demonstrate the plausibility of it, they are in fact examples and direct demonstration of it not only being possible, but actually happening. Important to the debate is the definition of multicellularity to distinguish mere clumps of cells from so-called colonies of single-celled organisms. In this 2019 paper, authors discussed the three main requirements of multicellular organisms: organization of space, growth, and cell differentiation, the former being an enabler of the latter two. They even cite Volvox carteri as an example of multicellular organism, which is one of the species of algae that we'll be discussing today. So this is the definition of multicellular I'll be working with throughout the debate, so there's no ambiguity. Multicellular Chlamydomonas reinhardtii display a novel two-stage life cycle in which motile unicellular propagules disperse shortly after transfer to fresh medium, then undergo successive rounds of mitosis to form non-motile multicellular clusters bound by an extracellular matrix. And if I'm not mistaken, I believe I skipped a slide. So I'll go back a little bit. So scientists are aware that biology doesn't care what we call it, and there are always going to be thousands of tiny deviations in the categories we try to assign. So real scientists recognize the multicellular individuality is on a spectrum, and it's not a categorical morphology. So still, multicellular algae meet the scientific definition I gave, and scientists call them multicellular. This is what the species in question does. The main paper in question, the authors clearly state that the algae, C. reinhardtii, evolved in their lab, possesses a novel multicellular life cycle. And to clarify, this does not mean that it's sometimes multicellular and sometimes not. It means that its life cycle, the entire life cycle, is multicellular. So they do shoot out unicellular propagules, they're called, which is how a new one is born. But just like humans begin as single cells, both before and after fertilization, this is all part of a multicellular life cycle, and it never reverses and goes back to unicellular. Other experiments, similar experiments on algae, have mutations that can limit the cell body number to specific numbers, including this one. In the main experiment, they're limited to 100. This is the main experiment we're going to be talking about. So this shows genetic control over body size, not haphazard, unrelated individuals sticking together. C. reinhardtii is in the wild, will clump unrelated individual cells together under times of stress, but these experiments demonstrate something totally novel: permanent and inherited change to the life cycle. The world of algae gives us a fascinating look into the evolution, since every step of the transitional form is preserved in living specimens. Now, evolution skeptics are unable to provide any criteria for whether or not these are the same kind of algae, whether they're related or not. Usually attempts to do this end up either overcrowding the ark or putting humans and apes together. Actually, exclusively. Either one or the other. But given they are microevolutionary steps away from each other, there's no reason to presume they're separately created kinds, and given we can observe these transitions in the lab, we have reason to believe they're plausibly related. To explain, is there any objection creationists can proffer to the microevolution of, say, the Eudorina species with gonidia, which are the cells making up the sphere body, to differentiate some of those cells as dedicated reproductive cells, and from that species with dedicated reproductive cells to a Volvox-like species with those dedicated germline cells that are localized full within the outer gonidia? No, these are trivial changes. So one question I have is, why would God make organisms that fill every step of transitional forms that evolution would predict, then expect us to reject the science of evolution? He would need to be a pretty stupid God, indeed, to sabotage himself thusly, and I don't think you believe in a stupid God, and neither do I. So the same gene that's responsible for regulating multicellularity in the wild-type multicellular Volvox is found in the unicellular wild-type C. reinhardtii, as predicted. This gene is integral to suppressing the division of somatic cells in the multicellular Volvox and evolved multicellular C. reinhardtii. And so asking why this happens helps us understand how it happens. Cooperation between cells necessarily means that some cells don't get to reproduce, at least in the multicellular way. And some cells are less fit and cannot survive alone while making the whole organism more fit. So this gene, Reg A, is integral to suppressing the division of somatic cells in multicellular volvox and evolved multicellular C. reinhardtii. So asking why this happens helps us understand how it happens. Figure A shows the regulatory gene of a unicellular individual that stops reproduction, turning on and off depending on timing and environment, which increases survival by putting less effort into reproduction. Figure B shows the same gene in a multicellular algae that responds to developmental cues. So in multicellular individuals, this makes some cells reproduce and others somatic and non-reproducing. So this demonstrates a really important evolutionary concept that creationists simply, I don't see them understanding. How new information can develop simply by minor alteration to existing traits. A gene that stops cell division can be implemented to create multicellularity through diversification of cell function by making some cells somatic, i.e. non-reproductive cells in the body of the algae, and some cells germline, i.e. reproductive. So this is the beginning of tissue and differentiation, and new information and functions from existing genetic information is observed. The expression of the same gene can lead to different functions. This is the basis of evolution. It's repurposing existing information. So I've demonstrated the functional organization of space through the building of limited number directly related multicellular bodies held together by an extracellular matrix, growth of this body which happens in all conditions, and cell differentiation between the germline and somatic cells. All these traits are lacked by the unicellular ancestors, thus multicellular evolution has been observed absolutely. And we can go into some of these quotations that I displayed on screen because I've gone through it pretty quickly. But that is indeed what is on the screen right now is indeed what is in the literature, specifically this experiment down at the bottom, which is the main experiment that's in question here. Chlamydomonas reinhardtii. And these are all three of the basic requirements for multicellular life. The original cells did not have these traits. The evolved cells do have these traits. Therefore, I have met my burden of proof. Well, the scientists who did the experiment have. Additionally, it seems individual cells can't survive alone because when you separate them, they're destroyed. Again, proving cell differentiation. And I've got a little bit of time, so I guess I'll go back and explain a little bit more. So the way these reproduce is that they shoot off a single-celled motile, meaning it can swim around, propagule, which is just a single cell in this case. It's not always in life, but in this species it is. And this cell will divide and they will stick together. They are all related. They're all daughter cells. None of these are just randomly sticking together with other colonies. These are all come from the same parent cell and they stick together. And the only ones that leave are ones that become another body. And they shoot off in these single cell differentiated propagules. And they're stick together with an extracellular matrix, which in wild type, when they end up doing this, it's either just sticking together unrelated individuals, or it's they're all clustered within the same cell wall, which is not the case here. So this is how they did it in this experiment. And I've got about a minute left, so I'll just show again the three criteria for multicellularity have been met by this experiment. And there are a lot of other experiments that support the same type of evolution and similar parts of the picture, different steps, similar steps. Yeah.
Okay. Thank you very much there. Snake was right for that opening statement. Roughly 12 minutes here. So let me restart the timer. Jamie, from Studio 215 Official, we're going to hand it over to you now. And whenever you're ready, the floor is yours for 12 minutes. And just make sure to unmute yourself, Jamie, when you're good to go. And the floor is yours.
Okay, I thought you were going to be the one unmuting me. So I'll go ahead and switch over to my presentation as well. I took notes on what you said, but I figured that when we get to the back and forth discussion, you'll bring up your slides again. So I'm just going to show the presentation I have so that our viewers have a full understanding of Chlamydomonas, because a lot of the things you mentioned are very well understood within the world of botany. However, these evolutionary papers you're referring to are just misrepresenting the already understood data. So let's go ahead and go through that. And then when we get to the back and forth, we can go through all of your slides and kind of explain everything that you went over. All right, so I'm going to try and go through this as fast as possible, but there's a couple things I might skip because there's a few things he talked about that I do want to get to before we get to the open discussion. All right, so for this debate, I want to expose the lies of evolution, teach you how to see through these lies for yourself, and use science for its intended purpose, which is to give God the glory for his creation. As far as Chlamydomonas reinhardti, here's a couple pictures if y'all want to go back and take screenshots of this for the anatomy. Chlamydomonas reinhardti is a single-cell algae that uses photosynthesis. It has up to 500 species. Volvox is not one of those, and Volvox is not anywhere related to it in the way that he's explaining, but we'll get into that a little bit later. The organism is a model organism because of its fast generation time, and I don't believe he mentioned this, but it does produce sexually and asexually. So that's one of the things that's going to be important as we move forward in this presentation. The ecological importance of this is that the Chlamydomonas reinhardti is one of immense importance to humans and nature alike. It can be used as a natural food source, fertilizer, and so much more. In the water, it can do anything from feeding fish to helping with decomposition in lakes, oceans, and sea sewage plants. On land, it is a great food source for a large variety of plants and animals because it is rich in vitamins, minerals, and nutrients. So the fact that it has multiple ways to reproduce just shows that it was designed for that purpose because it is crucial to our environment. It's obvious that Chlamydomonas reinhardti plays a crucial role in keeping our ecosystem alive. Luckily, God is such a great designer and has created this algae in such a way that it can survive and thrive in just about any climate, no matter how harsh or ideal it may be. Now, this is kind of the important part, is the reproduction process. So hopefully I'm not going too fast for the viewers, because this is where all the confusion that he's throwing out there is coming from. So there's 6 forms of reproduction that I've been able to find, and within some of those forms, they have off branches as well. Asexual reproduction and sexual reproduction. If conditions are unfavorable, they will switch over to sexual reproduction. In most situations, they like to do asexual reproduction, but they found that when they're in unfavorable conditions, that's when they actually start forming together and forming these clusters that he's talking about. We're going to go over that in just a second. So for asexual reproduction, the first one is zoospore. Sorry if the drawing is terrible. I scanned it on my printer. But this is the most common phase, so you can take a look at that if you would like. Here's the steps that go through that as well, but because we're pressed for time, I'm not going to go through every single step. However, if you want to go back over that in the future, feel free to do so. A planospore is going to be one of the important ones because, as you can see over here, when we look at the zoospores, when they start getting into harsh conditions, they can actually switch over to a hypnospore phase. And that could be one of the things that he's looking at. But I don't think it's a planospore that he's misrepresenting here. And then there's the steps for the planospore process if you want to go through and read that in the future as well. Now, Palmella, this is the one that I think is probably the best candidate for what he is misrepresenting, and what the scientists that are doing these peer-reviewed papers are misrepresenting. However, this isn't guaranteed. So if you look here, here's the zoospore process, right? This is going to be the normal process. When it gets into the unfavorable conditions, this is what it then does. It still has the replications inside of themselves that it had in the zoospore process. However, it builds a cell wall around itself. We're going to get to a more detailed graph in a little bit here, and it's essentially called aggregation. He should know about this since he went to college for this. However, I feel like he's probably just forgetting to mention that information because it's identical to what he shows in these papers. There's the steps for the Palmella stage. Again, you don't have to read all of this right now. I'm just trying to breeze through it so that we can get to the parts that I really want to cover. Then we get to the sexual reproduction phases, and this is probably another prime candidate for what they're misrepresenting in their peer-reviewed articles. So here's the isogamy. There's the images for isogamy. And we're just going to kind of go through some of the steps here. The fusion of the gametes, which are similar in size, shape, and structure, take place. The two gametes merge by their interior ends, and fusion begins. Remember that this is the part where if it's in harsh conditions, it'll start using these methods as well. So isogamy is one of the methods it'll use during harsh conditions. Anisogamy is another one. The images look similar, but they're different steps inside of that cycle. So that's why their pictures are somewhat similar. However, what we're looking at is we're seeing the parent cells split off into these gametes, then they fuse and create zygospores, or they can go over to a zygote, which then goes back into lateral fusion. And I'm sorry, I'm misrepresenting this. The parent cell can split off into one of two, the lateral fusion, which is on the left side, or the apical fusion, which is on the right side. So here's the information if you want to go through those steps. Again, I'm trying to get through this because a couple of the arguments he used is at the end of my presentation. So I want to hurry up and get to that part. Then we have oogamy, which is similar. Again, it's going to be going through the same processes, but the graph on the left-hand side is the detailed process for oogamy. And then on the right-hand side is just the general transition through the sexual phase. And then there's the steps for oogamy as well. So here's the part that I wanted to get to. This is an image of Chlamydomonas reinhardti during an ideal reproduction phase. So this is actually what it would look like while it's in its reproductive process in ideal conditions. This is less than ideal. This is less than ideal situations, and it forms these clusters, and this is known as aggregation. Now, if you look at all of the articles that they talk about where, oh, it proves evolution. When you look at those articles, these are the photos it's showing. I'm sorry about the blurry image, but they're just showing aggregation. This is known to be part of the sexual reproduction phase. So when it is in less than ideal situations, it forms clusters known as aggregation. In harsh conditions, these clusters can build a wall around them to help them survive until conditions get better. As you can see here, this is the image of them forming together, and then they build a wall around themselves to protect themselves in harsh environments. Now, what I noticed is some of these peer-reviewed articles will mention that they put them in conditions that favor multicellularity, but they never give you details of what those conditions are. I'm fairly confident that those conditions have something to do with biofertilizers or macronutrients, both of which contain nitrogen and would force these Chlamydomonas reinhardti cells to go into this process of basically trying to survive by creating this form around itself. The main term is aggregation, but I'm not sure if that's the only one. But here's the chart on that. So this is a chart showing some of the recorded social behaviors of Chlamydomonas reinhardti during stress. As you can see here, whenever it gets stressed, it's going to go into this aggregate state. And this aggregate state is actually what they're showing pictures of in their peer-reviewed articles and saying, this is it becoming multicellular. No, it's not, because what happens is once the conditions become more favorable, it then splits off from this aggregated state and goes to a palmelloid state, or palmella, which we talked about a second ago. And then from there, it goes right back into its normal reproduction phase and spits off into single cells. One of the peer-reviewed articles that I am going to be going over in a little bit here, that's exactly what they admit to in the article. And then they follow up that same admitting of it going back to single cell and saying, okay, that's it. It's multicellular. Just ignore that last part. So in comparison, this is the picture of their evidence for evolution. If you look down here in this corner, this is what they say is the multicellular Chlamydomonas evolution. However, again, let's go back and look at that again. Here is the aggregate state, right? This is the aggregate state. These are still single cells that are just trying to protect themselves in harsh environments. Then you go back over here and look at what they have as their evidence. It looks exactly like an aggregate state. So aggregate state from stress looks exactly like their evidence for evolution. I don't think that they proved anything other than they put Chlamydomonas in a stressful environment, and this is what it does in its stressful environment. So they're only reconfirming what we already know about this in botany. However, the problem is that they're using this data and misrepresenting it to act as some kind of proof for evolution. All right, and then here we go again. This is a single cell Chlamydomonas in the palmello stage. And then let's go ahead and look at, because he wants to mention Volvox cardii, there's a complete difference here. If you look at this on the left-hand side, the Chlamydomonas, it still has its single cells individually within its cell wall. When you look over here at the Volvox cardii, it has a cell wall, and then it has cells inside of it that make it multicellular. So it's not the same thing. I knew he was going to bring up Volvox. And the thing is, every article I've read, the only way they connect Volvox cardii to Chlamydomonas is by using comparative anatomy on a smaller level. Also known as homology. That's basically just comparing similarities between two things and saying, because they have some similarities between them, all of a sudden this proves that they're related. It doesn't prove that they're related. I've heard Kent say this thousands of times in his debates, and I'm going to go ahead and reinforce this idea that just because something has something similar to another organism doesn't prove they're related. It could also prove that they have similar design features from the same designer. If it worked on one organism, why wouldn't God use it on another organism? I don't understand why that's so hard for evolutionists to understand. Everything they use for evidence for evolution, in as far as homology and comparative anatomy goes, it could also be used for common design. If a designer uses something and it works, why wouldn't he use it again? I know they hate when we use the inanimate objects arguments. However, I'm going to go ahead and go over that anyway. If you have a car with 4 wheels and those 4 wheels are made of rubber and it works, then the next company that steals that idea and makes their own car, they're probably going to use rubber tires as well. It's because it's a material that works. So again, this whole comparing different features is not proof of anything. It could also be proof of a common designer. So therefore, that really just needs to be thrown out the window if you're going to use that for evidence, because we can use that for evidence on our end as well. So here's the difference, and here's why this is not multicellular. The
30 seconds.
30 seconds, I'll be quick. The Chlamydomonas is stalling its reproduction process because it's in harsh environments. This is not the final product. One condition becomes more ideal in the reproduction. Once conditions become more ideal in the reproduction process, it will continue to split into single-cell organisms again. He wanted to say that it never goes back to multicellularity. And if we have time to get to the peer-reviewed papers that I have, they admit that it does exactly that. As soon as it gets out of these harsh conditions, it splits back into single-cell organisms. So you're either ignorant to that fact or you're lying directly to the people to deceive them. I'll go ahead and close at that one.
Okay, Jamie, thank you so much for that opening statement. Gentlemen, great work, great job. Visuals, you're both coming in swinging and have obviously done a lot of prep for this. So, of course, I appreciate that. And that concludes the opening statement. So, Snake, Jamie, thank you so much. We're now moving into the 8-minute uninterrupted rebuttal portion of the debate. And so, Snake, whenever you're ready, we will hand you the floor. And you've got 8 minutes. I'll give you guys a 1-minute warning when you get to the 7-minute mark. That way you know to start winding things down. Okay, with that, floor is yours, Taylor. I'll put you full screen.
All right, so starting off with the Volvox issue, the reason I brought up Volvox is, well, not comparative anatomy, and that's not how the scientists compare it at all. It's comparative genomics, which is very different. Right. So the only reason I mentioned it is because of the comparative genomics, which is the fact that they have the same gene that can regulate division of cells. So what they're testing in these experiments is, well, at least the Reinhardtii one is, well, not the Reinhardtii one, this one that's on the screen. What they're testing is for whether or not the hypothesis of, can a gene that's just a normal survival gene be used to become an altruistic gene, which is that it some of the cells take a fitness hit, but the overall organism becomes more fit. So yes, they found that the social gene that downregulates cell division can be co-opted to basically, so in a normal, like the Volvox cartieri, this gene is being used to have multicellularity. It is essential and sufficient for this trait. And the same gene is found in the unicellular C. reinhardtii. So I'm not even arguing that they're related in this at all. I'm saying that the same gene that's present in the unicellulars is being used for the multicellular traits in other species. So yeah, common designer, apparently this common part can be used for either or. So that's the point. Moving on to, so this pretty much went exactly how I predicted it would go. Most of what you presented was pretty much irrelevant. So, well, I'll end with the best part, but for now, I noticed that you said science's intended purpose is to glorify God, when it's actually the complete opposite. Well, opposite depending on how you use the word. It's certainly not to denigrate God, if you're thinking that's what I meant. But it shows you don't understand the purpose of science. The purpose of science is to build knowledge of the natural world. God is the supernatural world, so they don't really have anything to do with each other. And even if you're saying, well, God is the creator of the natural world, well, the intended purpose is to learn about the natural world. That's it. You can do whatever else you want with that. People do good things, people do bad things. It's intended only to learn stuff about the natural world. So moving on to the aggregation and palmella phase. So you said it just shows aggregation. And that it's stalling the reproductive phase. So this is not true at all, because they don't go back, because they put them in the same cultures as wild type C. reinhardtii, which form no such clusters, and the multicellular algae continues to form obligate clusters, meaning it doesn't matter which environment you put them in. This is how science works. We test all the variables. We tested what your claim was, and your claim failed the scientific test, because we put them in the conditions that you said that they would go back to, and they didn't. You would know that if you read the paper. So you said that these clusters are identical to what's shown in these papers. This is the main paper that you said you wanted to talk about. It literally says these palmalloids differ in 2 important ways from the multicellular clusters observed in the experiment. I wonder if you read that far. It's fairly far into the, if I remember, it's fairly far into the paper. Maybe I forget exactly where, but if memory serves. First, palmalloids in wild type reinhardtii form plastically in response to conditions. So that means back and forth. Their clusters formed obligately, which mean, and they tested this in the same conditions in which you say they'll go back. This was also genetic, and they tested this as well. It is inherited, so it stays with them. Their children have this trait. They retain this trait in conditions that you say they'll go back. Second, the morphology of our multicellular clusters is distinct from that of palmalloids. So that's morphology. You just looked at it and kind of went, yeah, they look kind of similar, without performing any of the other tests detailed in this paper. Palmalloids typically consist of 4 to 16 cells. And yes, they are all within the mother cell wall. So, and that's why they're limited to 4 to 16 cells, because the cell wall will rupture if they get too large. In this, their morphology does not have a cell wall around all of them. They each have their own cell wall. So just looking at it and saying they look the same, it's a bunch of green globs, that's not good enough because you missed that major, major point. In contrast, these can contain more than 100 cells, and they don't have their cell walls. They're held together by an extracellular matrix. So completely different morphology, and completely different life cycle, completely in the same conditions. They're obviously related. That wasn't in question. So you said, quote, I quoted you. They're identical to what's shown in these papers. We just proved that wrong. Quote, it's just showing aggregation. We just proved that wrong. You say they never give details about the harsh conditions. They do detail those. I can, get the exact quote. It's not on screen right now, but so yeah. The final point I guess I'd like to make is the common design point is you can't use common design equally as comparative genomics because common design makes no novel scientific predictions. It's not scientific. The dual nested hierarchy, the fact that genomics predicts the nested hierarchy of morphology and vice versa, the fact that these predictions can actually predict genetic similarities in function and structure and location would not be expected in common design. So yeah, you can't use that evidence.
Okay, Snake, thank you so much for that 8-minute rebuttal. We're now going to hand it over to Jamie. Jamie, whenever you're ready, you also have 8 minutes. And on your first word, I'll start the timer.
Okay. Yeah. So the first thing I want to say is, I did misspeak and I said comparative anatomy when I meant to say comparative genomics. I was running through it fast and, I just am used to saying comparative anatomy. So my apologies on that. Yes, I was referring to comparative genomics, and I will go over that in just a second when we look at the peer-reviewed papers. Now, you wanted to mention the Reg A-like gene, and you said that this is why Volvox is multicellular, and therefore, because it's inside of the Chlamydomonas, that proves that it could become multicellular. Well, that's not exactly accurate because you don't know that's why the Volvox is multicellular. I see this all the time with evolutionary genomics studies. Is they always want to, grab a gene and say, okay, this does that and this does that, and then guess what? We find out 10 years, 5 years, 2 years, sometimes a couple months later, we find out that they were wrong about the purpose of that gene. So you can't come here and tell me that you know the purpose of every single gene and you know that that's exactly what it does, because over time we constantly find out that these genes actually do other things that we had no idea about. I mean, you can ask Standing for Truth about the ERVs, and everyone thought that they were useless, and then we find out that they have purposes. So I'm not going off topic. I just wanted to use that as an example to show you that you don't always know what those genes mean or what they do. Now, as far as you say that this gene, the Reg A-like gene, is the one that makes Volvox multicellular, I would ask you, how do you know that? Can you prove that? Can you show me that in the lab? Not just on paper. I want to see some kind of experiment that shows that if you remove that gene, all of a sudden it will no longer reproduce multicellular. Another thing I wanted to point out is you said that the multicellular clusters from Chlamydomonas, they cannot go back to single cell once they form these clusters. Now, I'm not going to argue the specific details behind the difference between your experiment and what aggregation is, because honestly, I didn't research that deep into that. However, I did research and read these peer-reviewed articles, and I'll go ahead and show you a couple of these peer-reviewed articles where they say that it does actually go back to single cell. So you can say that I'm wrong all you want, but your articles say the exact thing that you're saying is a lie. Let's start with slide 55. On slide 55, it says, in our experiments, we find that simple multicellularity can evolve rapidly in C. Reinhardti. Of course, they have to say that, otherwise they won't get approved by the journal editors. Like most extant multicellular organisms, we find that multicellular Reinhardti, which is not actually multicellular, I would argue that it's in the aggregate or the palmello stage, possesses a life history that alternates between uni and multicellular stages. So right here, they're trying to say it as discreetly as possible, but they say that it can switch back and forth between uni and multicellular, which you just said does not happen ever. So again, that's a bold-faced lie out of your mouth. Let's go to another slide. I'm going to scroll down here to slide 62. This is a different peer-reviewed article. I'm trying to go through this as quickly as possible. It says, every 3 days, they centrifuged each culture gently and used the bottom 10th to found the next generation. Since clusters of cells settle faster than single ones, this meant that they effectively selected for algae cells that have the tendency to clump together. So what this is saying is that they are artificially selecting the ones that they know will clump together more than likely won't. So they're selecting the ones that already clump together. And then when they clump together, they're saying, oh my goodness, it proves multicellularity. But then let's look at the next paragraph in this article. I didn't copy and paste it all because I was trying to put this presentation together pretty quickly. But in the next paragraph, it says, after about 50 transfers, the algae cells into one of the 10 cultures began to form clusters. Okay, so they selected it to form clusters. Now it's formed clusters. However, the cells stuck together for hours while they were settled from the centrifuge. Then they quickly broke apart into single cells again, each of which then divided to form new multicellular colonies. Now, what they're saying here is it split off into single cells after it was already a cluster, which you said they did not do. So there we go, finding out that you're actually either being ignorant to the fact that they can go back to single cell, or that they I'm sorry, I'm going over myself. But yeah, you said that they will not go back to single cell and they won't survive if they do. However, your own peer-reviewed article says this directly. If you want to know exactly which peer-reviewed article this is, this actually comes from New Science, and it's by Bob Holmes, and he just takes the information from the peer-reviewed articles. And I believe that the peer-reviewed article he took it from was, no, not that one. Sorry, I'm trying to find all of these. This one. He took it from the National Library of Medicine. Now, what's interesting about this one is, of course, they're going to start the article by saying that green algae whose lineage diverged from land plants over 1 billion years ago. So they have to add that as their first line of evidence. The reason for that is, first, they have to show their allegiance to the evolution cult that controls the peer-reviewed boards. Second, they must set the stage and enforce the idea that supports evolution so that as you read through the data, that's the only conclusion you will consider. And that's exactly what you did, because you know all about aggregation, you know all about palmello stages, and yet you completely dismiss those because they said that it was somehow different. But yet they don't explain how different it is. They just said that, I can't remember the exact terminology, but the cell wall was different. That's what they basically said. The cell wall was different, but it's still single cells clustered together. A cluster doesn't make it multicellular. I mean, you can pull up the definition of multicellular and we can go over that in our open discussion, but just because they cluster together doesn't make it multicellular. And your argument is that because they can't split back into single cells, that proves that they are now a multicellular organism. However, I just showed you in slide 62 and 63 that they can go back to single cells, and then they start forming another cluster, or they try and change the name to colonies, but they start forming more of these, which is basically meaning that now that they have already started this process of reproduction, that's the reproduction process that they are going to go with in the future. Remember, they have 6 different forms of reproduction. So of course, if they have been doing this for 10 to 50 cultures, more than likely they're going to go through that same reproduction process on the 51st or the 52nd, because they've already adapted to using this process of reproduction in whatever environment they're put into. Again, you're not mentioning any of the environments that they are put into. There's plenty of articles that say they're put into favorable conditions. Let me see if I can find one of them.
1 minute.
It did not. Okay, no, it's not this one. It's not that one. Okay, so I think it's right here. Nope. Oh man, did I take out that slide? There was a slide on one of the peer-reviewed articles that says they put it into conditions that favor multicellularity. I can't remember exactly which one it is because I'm kind of jumping all over the place to answer your questions. But yeah, what does that mean? That actually, I think it's this one. No. Okay, so I'll go ahead and scrap that because I'm all over the place at this point. But yeah, So you're saying that they're not the same reproduction cycles. However, if you look at it, they have dozens of different reproduction cycles. So which one are you saying they're not the same of? Remember that if we go back up here to the stress conditions, this isn't part of any of the 6 normal reproduction processes. This is what it does in an emergency situation. So if it can adapt to its environment and go through an aggregate state and then go back into one of its normal 6 processes, then how are you to say that what you're witnessing in the lab is not just another reproduction cycle that we don't know about or that we don't have details about? I'll go ahead and close it out on that.
Okay, Jamie, thank you so much for that 8-minute rebuttal. Gentlemen, that concludes the opening statements and the rebuttals. Again, great job and work so far. Lots of excellent points on the table to discuss. So with that, I am going to set the timer. I'm going to say between 40 and 50 minutes, depending on how the conversation is flowing. I do want to make sure there's ample time to discuss each point pertaining to this important topic. So Jamie just ended with his 8-minute rebuttal. Therefore, let's have Taylor, let's start with you in terms of first question or first point, or however you'd like to start off this discussion portion. So gentlemen, the floor is yours. Go ahead.
All right. So you said that how do we know that it's not just some other reproductive cycle going or staying in an aggregate life cycle? So the way we know that is, I already told you and read from the paper that they tested that specifically this variable. They put them in the same conditions as wild type C. reinhardtii that do not form these clusters, and they still formed the clusters. So they're genetically distinct. So how is that not answer your question?
So again, I responded to that exact statement. If they've already been doing this process for 50 generations, then why wouldn't they continue that same process? Because that's what they've become used to. Now, I know that they may put it back into normal conditions, but if it took them 50 generations to get to that stage of of reproduction with the aggregate and then palmella or whatever you want to call it, then why would you expect it to automatically go back to its asexual process?
Because they know how this works. They've tested it. They test how long, the conditions in which they go back. So they're not used to it. This isn't an individual that's been there and it's, acclimated. It got a tan and it's just used to it now. These are generations past. It's ingrained in them genetically. That's why they don't go back.
So you basically said that the reason we know it is because we know it. And that's not a sufficient answer.
No.
That's exactly what you just said.
No.
Yeah, you
gave us examples of the conditions in which they flip back and forth. We know that they do this. Mm-hmm. That's not me just saying we know. We agree on this already. And they put them in conditions where those original species, the wild type, will go unicellular. And they left them there. And they formed obligate clusters.
Yeah,
but you're just saying, well, they're used to it. That's not a scientific term. That's not an argument.
Yeah, but you just saw the part of the paper where I showed that after they form these clusters, they then immediately branch back out into single cells. Do you want me to pull that back up for you?
What you quoted was, yeah, go ahead.
Here we go. I think it's this one. Yeah, so let me go ahead and switch that over. I don't know that. No, it's too low. Okay. So let me go ahead and go to full screen for them. It says right here, sure enough, after about 50 transfers, Algae cells in one of the 10 cultures began to form clusters. To the researchers' surprise, these clusters seemed to pass through a coordinated life cycle. And then here's the key part of this whole sentence, or this whole paragraph. Cells stuck together for hours while they settled from the centrifuge, then quickly broke apart into single cells again, which is what you just said they wouldn't do.
Mm-hmm. Here's the problem: is you're quoting from a different paper.
It doesn't matter. It's the same. They're doing the same researches. Just because you're one example.
It's a different experiment and a different line, a different genetic line. So you can't use that to talk about this genetic line.
Yeah, but what's
the other thing that you quoted?
in the paper that you're talking about, they may have not mentioned that it could split back into single cells. How long were they waiting before they determined the final results? Did it say how long that these generations continued in these clusters?
They had at
Or
least
did they just say,
73
oh, it's done, that's the end of the
generations,
paper?
if memory serves.
Okay, so 3 generations, and then what?
73.
Okay, so 73, and then what happened after the 73rd? What happened on the 74th generation?
They kept, I think they went past 73. I'm searching through the article. But that's just the one I remember.
Yeah, so you
and
can
there's
send me
like
that
48
article, and
hours between each cycle.
yeah, because you can send me that article. I'll read through the whole thing and, add that to my presentation, because every single article I found, eventually it goes back to splitting into single cells. Now,
And that evolution can do that. Why wouldn't it?
Well, here's my thing though, is that you're seeing all of these single cells cluster together, which can be replicated in other forms of reproduction. And you're saying that, oh, because it had a different cell wall or whatever
They're
your
not,
argument
it's
was
not
on
a
that.
different cell wall. And this is novel. This cycle has not been seen in nature before at all. It's novel. And it's not a different cell wall, as you said. That's something I wrote down. You said they didn't explain how they're different, and I quoted it to you how they're different. They each have their own cell wall instead of sharing a cell wall. That's a morphological difference that you ignored in your opening and you ignored in your rebuttal.
So, all right. So since you said I ignored that, I must have just glanced right over that. So if we look at this chart right here for cells, these single cells have their own cell walls inside of this. And then outside of that single cell, they have another cell wall that's protecting them from the harsh environments. That's what I'm claiming you're misrepresenting.
That's not a cell wall. The other thing that you quoted from this, actually from this paper this time. Okay. That, I think that's the extracellular matrix, which is not a cell wall. But you did quote from this paper, actually. You said, in our experiments, we find simple multicellularity can evolve rapidly in C. reinhardtii. Like most extant multicellular organisms, we find multicellular C. reinhardtii possesses a life history that alternates between uni and multicellular stages. You took that to mean that it will go back to unicellular and start reproducing unicellular?
Yeah, but see, here's the thing. Right? They're saying that it went back and forth between uni and multicellular.
But that's not what that's saying.
That's exactly what they said in the paper.
No, it's not.
Exactly what they said in the paper. That's it right there. It says the life history
that's one life
between
cycle.
uni and multicellular stages. But the thing is that that doesn't just because they're clustered together. Okay, so let me ask you
this. Can I explain why you're wrong about this?
Okay, let me ask you this, and then you can add that to the end of your answer.
Well, I don't want to move on from this.
Okay, yeah, we're not moving around from this. We're staying right here on this topic. Is a cluster or a colony considered multicellular?
Not necessarily.
Okay, well, that's all they're showing. They're just showing clusters and colonies.
No,
in every single one of the papers I have 4 different papers in this presentation. Those are the only ones I could find on Chlamydomonas and evolution. So I'd love to see your papers so I can also look at those, because every single one of them says that it formed a cluster or a colony. And then you're taking that to say that it was multicellular, but you just said
all of the scientists are also saying that too. And because it meets the definition, because it's a cluster of differentiated cells. So they have reproductive differentiation and motility differentiation. So that's 2 levels of differentiation, which means it's multicellular. That's the beginning of tissue differentiation. So what you have up on your screen is talking about the stages of a life stages of a multicellular organism. So as I already explained, they shoot off single cells that become multicellular. That's it. That's all they observed from after this type was established. And this is not different from any other multicellular organism which reproduces with a single cell That every single organism starts as a single cell, including this one.
So what
So
you're saying
the
is
unicellular stage is when it's reproducing. It shoots out its reproductive, differentiated, motile cell, which then becomes multicellular and stays multicellular.
And see, this is where you're misrepresenting, because it shoots off that single cell, and then that single cell goes and forms more clusters. It doesn't form a multicellular organism. It goes and connects to other single cells that shot off.
No, absolutely. That is purely wrong. They're all clonal cells. They're all daughter cells of that single original cell that shoots off. They don't join with others. You need to read more carefully.
Okay, so let me go ahead and go back over here then. So, zoospores. Let's pull that image up one more time. One little organism right here is going to form into two, and then form into four, and then those four are going to branch off. This is what you're witnessing. You're witnessing a zoospore, and you're saying that it's now creating a multicell. You
No,
could also
because
be
that-
witnessing this. You
No,
could be
those
witnessing
share-
a planospore.
Those share a cell wall. So no, try again.
No, they don't share. They don't share a cell wall.
This is why this life cycle is novel, because nothing you show is the same.
Okay, so what you're telling me is that these single cells are shooting off, and then they're just magically creating more within themselves, and then shooting off single cells again, as if you're trying to compare it to the sperm of a human going into an egg, using the DNA of the egg to then form a baby human. That's not the same thing at all. They're going in, they're just reproducing in one of these other 6 forms, and you're trying to say that that's multicellular.
No, it's a novel form of reproduction. Actually, it doesn't match any of those 6 forms. That's what it says in the paper.
Well,
And
okay,
it-
that's
one
what
of
it
the
says in the
reasons-
paper,
one of
but
the
you look at the
reasons it's
diagrams,
novel-
and that's exactly what it shows. It
You
doesn't matter what
claimed-
they say, it matters what they show.
They say what's in the experiment. So they claim in the paper, these clusters develop clonally by daughter cells staying together after mitotic reproduction, not by aggregation of single cells that cohabit the same growth chamber. So that's what you said earlier a couple minutes ago is false. And yeah, they report what's in the experiment. People can redo this experiment and call BS on them. That's the thing about science. That's how they get papers published, because it's reproducible.
So what you're showing me is very similar to all of the other reproduction processes in harsh conditions. And then you're saying that they're not the same. But how are they not the same?
Yeah. Similar is
Please.
not exactly the same.
Okay, so how is this multicellular then? Explain that to me. How is your image on the left, that is basically identical to my image on the right, how is that not single cell clusters, and that's a multicellular organism? I want to know the technical terms for why this is multicellular compared to mine that is not multicellular, because they look identical.
I
So
mean,
please
you should
explain
have
that
read
to
the
me.
paper.
I don't
They
have that
explain it.
paper. The paper that you're showing, I don't
The
have
one I
that paper.
quoted that this debate's about, that you responded to in Kent's video.
No, my papers, I actually have all the titles of them here, and that's not one of my papers. But I will gladly go through that paper as well. Hold on one second. Maybe this.
So you didn't read it. That would be fairly important, because this is the one I cite all the time. This is the one I cited to Kent. I tried to go back and check, but Kent took down your video. He never put up my video.
Oh, okay. You know what? Yeah, you are right. I do have that. I do have that here. So yeah, This is the one that I was talking about. Okay. So I'm glad that you pointed that out, because this is what I was looking for earlier that I couldn't find. Let me go ahead and pull that up to large screen for a second. If it'll, yeah. Okay, cool. So right here, it says, we subjected the algae Chlamydomonas reinhardti to conditions that favor multicellularity. Okay, so what does that mean, that they put it in conditions that favor multicellularity? Did they use biofertilizers? Did they use macronutrients? Because both of those have nitrogen in them. And when they are put into nitrogen environments, they cluster up and then they go into the aggregated state. So what you're telling me is that they put them in conditions that they do not specify. You say that they specify them, but they don't specify them. Okay, so what are the favored conditions then? How is that a favorite condition?
In other experiments, they do predation. In this experiment, they did centrifuge.
No, that they did not say that in that paper at all. I scanned through that entire paper looking for centrifuge, and that's not what they said in that paper. The only mention they have of the environments is this one sentence that says they subjected the Chlamydomonas to conditions that favor multicellularity. I read through that entire paper, and they did not specify what those conditions consist of.
Quoting from the paper, to select for cluster formation, the first step in the transition to multicellularity, 10 populations were subjected to strong selection for rapid settling through liquid medium. Briefly, we performed the selection by centrifuging 1 milliliter of each population at 100 G for 5 seconds, and then transferring only the bottom 100 microliters to fresh medium. So they literally say that they use a centrifuge.
Yeah, and then what happens after the centrifuge? They clump together temporarily, and then right after they clump together, they quickly broke apart into single cells again. So there we go. They go back into their single cells.
That's from a different paper, but this is how evolution works. So if you subject a population to a stress, immediately they're not all going to just start morphing like a Pokemon into their next stage. They have to have children. So they subject them to conditions which favor multicellularity. So at first, yes, they are not genetically changed. They form these temporary clusters. But after several generations, they formed clusters that don't break up when you put them back in the wild conditions.
Okay,
because those ones survived better because they always clump.
Okay, again, it still says that over time they will go back into single cells.
It doesn't. That's a different paper.
It doesn't matter if it's a different paper. It's going to be the same result either way. It's going to turn into the same result. They're going to split off.
That's a different population.
Cells, and then once they split off into those single cells, you now have a back and forth seesaw. You're never going to get another organism from that. They may cluster together and form what you claim is a multicellular organism, but then over time it splits back into a single cell. How does that now evolve into a sponge? Or how does that now evolve into whatever the next stage of your fairy tale is? You see, you're watching a back and forth process, and it always goes back to unicellular. It's never made it past that stage. So you're seeing them clump together. You're seeing them form a cell wall around themselves so they're cell walls within cell walls like inception. And then you're saying, okay, well, now it's a multicellular organism. Okay, well, why doesn't it stay that way? It always ends up going back into single cell.
Which they also tested.
Okay, so let me see the genetic part of it then. Pull up the genes that have changed. I want to see a new gene added into that, because you want to mention
it's not a new gene added onto it.
Oh, so no new genes were added.
It's a change to an existing gene.
Okay, so one of those genes changed. That could have been
it's the Reg-A-like gene.
Right, and that couldn't have been
almost identical to the one that is necessary for the Volvox. And yes, they've tested that. The paper I cited, they cited someone else for that. But apparently that's been experimented on as well. So
okay, so let me ask this. Before the experiment, did Chlamydomonas have that gene?
Yes.
And then after the experiment, it still had that gene.
Mm-hmm. And it was mutated and it functioned. It was mutated and functioned differently to have a completely new trait. New information.
So what's to say that that's not part of the gene itself?
New information from existing information, because it was a mutant.
How do you know that's new information? How do you know you didn't just activate something that was already built into that gene? If this was a design code, how do you know that it doesn't have multiple purposes? Because we see that multiple times throughout science. We see that we think that a DNA, we think that a gene, for instance, ERVs, we think it has one specific purpose. And then later on, we find out it has multiple purposes. So how do you know that this Reg-A-like gene didn't already have that purpose?
If it was designed to evolve, because this type of life cycle has never been observed before this experiment. And as you showed, these species have been studied very intently to the point that we have all of these different life cycles that we've recorded. And this one has never been recorded. It's only been recorded in response to experiments like this. And it's different from all the other life cycles. And it remains when we put them back into wild type conditions, where its ancestors, direct ancestors, remain unicellular.
So you're saying that you take this single cell, you force it to go into a cluster that it now is staying into, you put it into nature, it survives, and it never goes back into single cell.
That's what was observed in this experiment.
Okay, I didn't get any of those results in that paper. So can you pull up the paper and show me that?
We've shown several times that you were unaware of what's in this paper.
Yeah, because I don't have all of them off the top of my head. Keep in mind, I've only been studying
this is what the debate's
for the past
about.
month. So yeah, keep in mind, I've only been studying this for the past month. So I don't have the entire paper memorized. I just took out, clips here and there. But I mean, I could pull up the whole thing.
I guess you should have prepared more.
Word for word.
That's not my problem.
Well, honestly, I thought that you were going to bail again. So you bailed the first time. I thought you were going to bail again. So I didn't feel that I needed to break down every single sentence of the paper. However, what you're saying what you're saying is that this single cell formed into a cluster that you're now claiming is multicellular, and then they put it into nature. It survived and never went back to single cell. But how long did they monitor that? Did they put any of that information in there? What happens to this multicell now? Every single one is going to be multicell from there on. Do we have a new species of algae?
That's what was observed.
Okay, so pull that up and show that to me then, since you have
I already did.
Pull it up. Let me see it so I can read through it. I want to read the wording on it. All right, so cultures of Reinhardti
they differ in two important ways from the multicellular clusters. The palmella is different from the clusters observed in the experiment. Then it gives the reasons why you questioned, why is it different? Well, it's very explicit in this paper. They form clusters obligately, which means always, in culture conditions, which wild type form no such clusters.
And how do you know that it doesn't form those clusters in the wild? They say that it doesn't form those clusters in the wild, but how do you know that? Because they're saying it's obligate. What's different about it? That's what I'm saying. You're not explaining what's different about it.
They grow wild type in those same conditions, and they don't form clusters.
If they're growing it, it's not wild. It's a controlled.
So you don't know what wild type means.
And then you're saying that these clusters
do you know what wild type means in biology?
Okay, so go ahead and explain it to me, please. What does wild type mean?
Okay, so this is like really basic. I'm not trying to like attack you personally, but if you're gonna have debates on biology, you might want to know some biology. So wild type, and this is one of the problems, is I'm not faulting you personally, but you don't know how much you don't know. This is
okay. So again,
Dunning-Kruger effect
about these multicellular
wild type.
Clusters, how do you know that it doesn't happen in the wild?
Wild type is a specimen harvested from the wild that is genetically identical to the ones that are living outside of labs in nature.
Okay, but as soon as you grow it in the lab, it's no longer wild.
AKA not mutated.
You're putting it under specific conditions.
No, it is still wild type.
Okay, so you're taking something from the wild, you're putting it into a controlled environment, but it's still wild.
So if you've ever been in the lab, if you've done mutational experiments on bacteria or on fruit flies, they'll use the terms wild type because these are not mutated. They keep the genetics the same. What happens when they mutate in wild type is that the mutation just gets cycled out because there's no evolutionary pressure. When you add evolutionary pressures to wild type, they start changing. The mutation spreads through the population. Now they're no longer wild type.
Okay, so pull back up your slide right quick because I'm pretty sure you didn't mention the question that I was asking you. So what is different about these multicellular clusters, and how do you know that that doesn't happen in the wild? Because you say that our multicellular clusters formed obligately in cultured conditions in which the wild type forms no such clusters. So how do you know that those clusters are different than the wild type clusters, and how do you know that they have not done that in the wild?
Well, one, it says it right here. They form obligate clusters.
That's what I'm saying, though. How do you know those clusters are different? Just because they... Okay, go on, please.
And over this many times, those clusters are different because, one, they don't have a cell wall. They have an extracellular matrix. They have a cell wall over every individual, not the entire thing. It says
so
this
you said
right
they
here.
have a cell matrix, right? Isn't that what you just said about the aggregate state? That's the same thing you said about the aggregate state. When I showed you the graph of the aggregate state, you said, I'm pretty sure that's just a cell matrix. So you're saying that it's not aggregate, but you're describing the same thing that you tried to debunk me on when I was showing you the aggregate part of the chart.
But the aggregates don't form obligate clusters. So there's that. So then the cell wall thing is explained down here. They're not held together by a parental cell wall. It's an extracellular matrix.
Right.
Aggregates are also not all the same daughter cells. Like these.
Right. So as they are reproducing, other cells are coming into them. Is that what you're saying? Because you're saying they're not the same daughter cells. Explain that.
Which them are you talking about?
Because you're saying that the aggregate doesn't have the same daughter cells. There's different cells in there. But if they are all if they're all one organism, then they should all be the same cells. They should all be part of the same daughter cells, unless they are forming clusters with other cells around them, which would explain the aggregation process.
Significantly, clusters develop clonally by daughter cells staying together after mitotic reproduction, not by aggregation of single cells that cohabit the same growth chamber.
Okay, but that mitotic reproduction, I'm pretty sure that was part of the sexual reproduction phase when I showed those graphs.
Okay, irrelevant.
So you're looking at sexual reproduction now? See, you're trying to blend asexual and sexual reproduction and say that that's multicellular.
No.
Yeah, because that's exactly what it shows in in the sexual reproduction phase.
Developing clonally from the mother cell is not sexual.
Right, but mitotic reproduction I'm sorry, it's not sexual. My apologies. It's asexual. So they're fusing, or they're going through the fusion process to create more of themselves inside of that matrix, as you say, which could be defined as aggregation. But just because they said it's not aggregation, that proves that it is multicellular and they've never seen it before.
No, they have not seen it before. As you said, there are multiple studies on the life cycles of these organisms, and this is never found in any wild type.
It's found when they're under stress conditions. That's what
not in the wild.
Comes from.
Not in wild type. This only happens after laboratory stress conditions in which genetic changes occur to the entire population.
So you are aware that these Chlamydomonas can survive in conditions much harsher than human environments. So there is Chlamydomonas in sub-zero temperatures that survive through aggregation. So how do you know that that's not exactly what you're witnessing here? You put it through harsh conditions that then made it replicate what has already been done in the wild, but you just never observed in the wild. In order to say that it's never been observed in the wild, knowing that Chlamydomonas can go through extreme harsh conditions, you have to now know that every single place that Chlamydomonas grows, it has never done that before. So see, you're limiting.
That's why science says it hasn't been observed. We're not saying it's impossible. They could have evolved in the wild. In fact, that's kind of
But they're not
The
evolving
thing.
though. They're just forming clusters to stay alive until they can finish their reproduction process.
They're
Because again,
forming clusters
in
with single bodies with limited cells that are all related. They're not unrelated, just bumping into each other. And they have cell differentiation, which meets the definition of multicellular.
right, but then they split off into single cells, and
Right.
then they have
Okay.
to go back through these processes again. So they never stay multicellular. They go right back to single cell processes.
No, they don't. They stay multicellular. They shoot off one single cell to reproduce. That becomes multicellular and stays multicellular. That's how everything reproduces.
But see, this
Through a
is where
single cell.
this is where we
Your
are.
father's sperm was a single cell that met your mother's egg, which was a single cell, and they formed a single cell.
Right, because it was designed that way.
So we go through a single cell phase. Humans do. Of a
I mean,
multicellular
if you
life
want to call it
cycle.
that, sure. But the thing is, that's
how
not
science
that's
words it.
like that's not exactly a single cell phase. Just because a single cell went in there, put the DNA code into the egg, and then that grew into a multicell, that's not proving single cell to multicell. And again,
It is. It's when your mother's egg is fertilized, it is a single cell.
right. But the
And
thing
so
is,
you go through a
that's
single
part of
cellular
the design
stage.
process. So see, now you're trying to do the whole Ernest Haeckel argument, which was debunked 100 years ago. So it does, just because we start as a sperm in a cell, just because we start as a sperm that goes into an egg, that doesn't mean that we evolve from a single cell into a multicell. That's part of the growth process. That single cell had all the DNA to create the multicell. It didn't magically learn to multiply itself. That's part of its design process.
It didn't magically learn. The changes were genetic, so they're not just used to being multicellular. They are genetically obligated to form these clusters.
Right. So what you're showing is just all of these replication processes, and you're saying, okay, well, it doesn't happen in the wild, even though we have it recorded happening in the wild.
We don't.
Okay, then where did all these charts come from?
Everything you went through,
processes.
Everything you went through is different, and the authors of this paper know that because it's been studied, and the word they use is a novel life cycle.
Okay, so that's what I've been asking you this whole time. What is different about this? All you're saying is that it's obligate. It's
I'm gonna have to
obligated
write
now.
this down for you.
What is literally different about it, though? It goes to when you look at the final product of what you call multicellular, it looks exactly like an aggregate or a palmetto stage. And then they branch off into single cells, which would then fit the graph of the palmetto stage. Eventually, it branches off into zoospores.
Nope. Palmella stage shares one single cell wall and is limited to 4 to 16 cells. These, in the experiment, can have more than 100 cells, and they don't share a cell wall. They each have their own cell wall.
Yeah, but so that's the same thing that was in the aggregate stage. They have their own cell walls, and then they have a matrix around it, which is what you just said. You said that, oh, that's just a matrix. Well, yeah, but that's exactly what you're looking at too.
But they don't form obligate clusters. So there's a difference in every single life cycle.
And see, that's where we're going back in this circular argument, because what about this obligate cluster is different than the regular aggregate stage? What's different about that?
I just said they don't form obligate clusters. Do you think the scientists just lied and got their
so you're saying
research through scientific peer review by just lying, even though there are other scientists that know about this stuff?
Yes, they do it all the time. You can see that with the entire I don't want to get off topic, so I won't go down that rabbit hole. But yes, I had a whole presentation on fossils and how they blatantly lie. So yes, you do see scientists lying all the time. Again,
well, you don't have a background in science, and you don't seem to read the papers you talk about, and you don't understand the scientific terms in the papers. So don't think that you are accurately saying when you found a flaw in scientific literature, because there are people with actual degrees who don't find the flaws in it. And no, they don't have to say anything about evolution to publish their research, which I find very frustrating because I'm looking for all this research to show evolution, and a lot of them just don't even mention evolution.
The very first
To my
sentence of every
chagrin.
peer-reviewed paper is millions of years ago, or every single peer-reviewed paper, you can pull up any peer-reviewed paper. Within the first paragraph, they have a whole doctrine of evolution that they're explaining. Every single
Some of them do, not all of them.
But see, you're deviating away from the question that I was asking, because it's funny. I said, what's the difference between the aggregate stage and these obligate? So I said, what's the difference between this aggregate stage and the obligate clusters? And you said, well, they're obligate. Well, what's the difference between that? That's literally what I'm asking you. What's the difference between obligate clusters and aggregate clusters? What's the difference between that?
The difference, it's kind of in the word, they're obligated to form clusters. They always form clusters, even in wild conditions where the wild type does not form clusters.
But then you also admit that they have their own cell walls and they're inside of a matrix. They never are one. They're never one organism. They're a bunch of single-celled organisms clustered together. And then I asked you earlier. I said, I asked you earlier.
Do you think humans have all their cells share one cell wall?
No, but we are one organism.
All of our cells are bound together with extracellular matrix.
Right, but we're not a cluster or a colony. We are a organism. Just like
because we have cell differentiation, just like this Reinhardtii in the experiment.
Okay, so it looks like we're just going in a circle here, because you're saying that these single cells molded together, and now you have single cells with their walls still separate from each other. And then you say that they differentiate, but they're inside of this cell matrix that looks exactly like the aggregate stage. And somehow when they split off back into single cells, they're just reproducing like humans would. But that's exactly what they do when they're single cells anyway.
I didn't say they reproduce like humans would. I said that humans also have a single-celled stage, which, because the paper says this has a single-cell stage, you thought that meant that they go back and forth between multicellular and single cellular.
Yes, because they don't. You see the other research papers that say they do go back to their single cell forms almost immediately. So just because
wild type does that, these do not.
That's not what it said at all in the second paper that I showed you. So you're taking one special situation where the multi-celled where the single-celled clusters stayed together, and you're saying, okay, that proves evolution. But then we see
this population evolved, not the one you're talking about.
Okay, but then did it survive in the wild? What happens if you separate those single cells? You said they don't survive. Where's the evidence of that? Did they put that in the paper? Because I didn't see that in the paper.
Did they what?
You said that if you try and separate the single cells, they won't survive. But yet that's exactly what they do. They separate back out into single cells to create more clusters. And then earlier on in the presentation, I asked you, is a cluster or a colony a multi-cell organism? And you said no. But then
I said not necessarily.
Okay, so is it a cluster, a colony, or a multi-cell organism?
Yes.
Yes. Okay, so it's all three of those. So a cluster is a multi-cell organism then.
Multicellular organisms are clusters of cells.
Okay, so see, this is where you get into, arguing definitions, because I mean,
there are more criteria than that. There are more criteria than that, obviously.
And that's what I'm asking you. What makes it different than what turns it from a cluster or a colony, which is the only thing they ever reported as in these peer-reviewed articles, into a multicellular organism? They say that these single cells formed clusters. And then in your paper, which I do end up having, they say those clusters are now a multicellular organism. So what is the line that jumps from cluster to multicell?
Are you ready to write it down?
Yeah, let's do it.
Limited cell number. The extracellular matrix that goes into functionalization of organization of space. Growth, obligate growth phase and clustering. Cell differentiation, motile versus non-motile, propagules, and germline versus somatic.
Okay, so I don't know germline versus somatic, but as far as the limited cell numbers, and what was the second one you said? The growth becomes obligate, so now it's going to create clusters as it produces more in the future. That doesn't prove that it's multicellular, in my opinion. And I'll go and read the article and, break it down into more detail, and I'll go look up because you're talking about that same slide that had the 3 criterias, correct? I think they're just listing characteristics of the clusters and then saying, okay, that's multicellular now. That doesn't sound like it's changing anything from a cluster to a multicell organism. I believe that a multicellular organism would mean that each one of those cells has a specific process that it goes through to keep the entire unit together. Is that correct? Just like how we have lungs that allow us to breathe, we have a heart that pumps blood, we have liver that filters out chemicals. Where do you see that inside of these clusters? How are they designating purposes between each other? And now
there's cell differentiation.
Okay, well, what are those purposes? You didn't specify any of those details.
I did. The propagules, which is the term they used for the reproductive cell that shoots off, is motile. The cluster cells that are somatic are non-motile.
Okay,
so
the
cells differentiate
when it branches off of reproduction, though.
They are motile, so they're able to swim away. The germline is able to swim away. So there's 2 differences. There are somatic cells, which make up the main body, and then there are reproductive germline cells. So that's cell differentiation. And there's a further differentiation because there's a difference in motility between those cells.
So what that sounds like to me is that some of them have been designated to go and reproduce more, and the other ones are designated to keep the unit alive.
Yes.
But that doesn't necessarily mean that that's now a multicellular organism. That could just be a survival tactic for them.
That is exactly what multicellularity is. Cell differentiation.
But they're still
of a
branching
single
back
body
off into single cells.
But they don't, because those single cells don't stay single cells. They all become clusters. Obligate clustering.
Right, but from the process of that single cell splitting off and then forming more clusters, what happens after it splits off? How does it go into forming the next set of clusters?
It loses its motility and starts dividing into a body.
Which could be the zoospore phase, the aplanospore phase, the palmella stage. So now it's going into asexual.
The zoospore and palmella, I know specifically, they share a cellular, a cell wall. So that's not true at all. This is a novel life cycle.
Okay, so I'll have to look into that a little bit more because I think there's something I'm misunderstanding in your explanation here. So I'll have to look into that a little bit deeper. But I'm not seeing how that makes it multicellular because there's still single cells within this one, I guess you call it a matrix. Am I correct? They're all inside of this matrix. All of these clusters are within this matrix. Some of them now designate themselves to go reproduce. The others are reserving their energy to keep the ones that are going to reproduce alive. That just sounds to me like a survival tactic of these single cells.
It's not. It's a survival tactic of the entire body, the entire individual. All these cells are cooperating as one body. That's multicellularity.
So I guess we need to make a more solid line between what is single cell and what is multi-cell, because to me, that doesn't sound like it's multi-cell. It sounds like they're this cluster that said, okay, now we got to figure out a way to survive. And this is what they decided to survive, is that some of them are going to go on to reproduce and the others are going to protect those that are going to reproduce. That doesn't necessarily mean, that's like saying that, that's like take a city, for example, like New York City. Okay. Not everyone is going to be able to, not one person is going to be able to run that entire city. They have to have certain people to run that, to run different departments of that city. And they've decided to come together as a group of people to keep that city running. That doesn't mean that the people are now one unit. Like it's it's a city, but there's still separate people within that city. That's essentially what you're saying to me, because let's look at these Chlamydomonas as people.
There's one difference.
Okay, what's the difference?
They're all daughters of the same cell. They're all genetically identical.
Okay, so let's say that a whole family.
Not, so there's single-celled organisms that come together, even of different species, and they stick together and they cooperate. That's not a multicellular organism. That's like a unicellular colony, because they're different genetic lines coming together. But these reproduce from a single mother cell and they stay together. It's one body. An extracellular matrix is just its proteins that bind these cells together. Same as
Okay, I
like
mean,
collagen or
so
something. That's an extracellular matrix.
okay, so what you're describing to me could all like take the same analogy, but then so I'm Hispanic. So let's say that, I have 15 brothers and sisters, and we decide to stay in the same house to support each other. That doesn't make us one person. We're still separate people inside of that house. That's basically what
But
you're saying is the difference.
this is the criteria for multicellularity in organisms. An organism that has multicellularity has its cells cooperating with each other. Does it not?
Yeah, it does, but they are
Okay.
designed for specific
So-
functions.
So
But
if
what you're
you're
looking at individuals that have decided to take a specific role to survive, and then they eventually split back off into single cells.
cell differentiation, they split
Oh,
off
your mic sounds
on
a lot better now. I don't know what you did different, but something sounds better now. Okay, go
Really?
ahead.
I haven't changed anything. My mic evolved, I guess. Yeah, so they split off, one splits off and becomes multicellular. But so to use your analogy, if you and your brothers and sisters all kind of butted off each other and you stayed stuck together, I would call you some kind of single superorganism.
No, the analogy is that my mother and father created 15 brothers and sisters, and instead of moving out of the house and going our separate ways, which is what these Chlamydomonas would normally do, they decided to stay together, maybe because of the conditions that they were in, or maybe because that's what they've been doing for the past 73 generations. And now that they're in this process, they're staying inside of these clusters, and they're saying, okay, well, this brother and this brother and this brother and this sister are going to go out and create more. And then they go out and separate from the body. And then from there, they now are going through one of the reproduction processes to create more. And then you're saying that cluster is now is an obligated multicellular organism.
But this is why biology is slippery. So yeah,
to say the least. Yeah.
Yeah,
there's a
you
lot of
can
gray
make an
areas.
analogy like that, but the multicellularity has a definition. There
Mhm.
are cooperating cells in one body. So these cells are bound together in one body,
Mhm.
and they have cell differentiation.
But what's
What
the
are
body
your
that they're bound together
criteria?
in? Are you saying that the matrix is now the body?
No, it's what's binding the cells together. The cells and the matrix are the body.
Okay, what's binding it together? That's what I'm saying. What makes it a multi-cell? Because so far you've just explained that they have their own separate walls. They have a matrix that they're within. So what's the binding agent that now makes them one organism?
Both.
That's aggregation. Like you can't use that as your
No.
answer when it's the same thing you see in aggregation.
No, because they're all clonal.
And then also you can look at the parent cell too. Like that's exactly what a parent cell looks like, except for the fact that the wall is it's an actual wall, whereas in your representation it's just a matrix. So I don't see where the fine line is that switches it from a unicell to a multicell.
They're all clonal. They're all one body. Their cell number is limited. They have obligate growth phase.
Through that same list. Again, that's
and
just
they have cell
characteristics
differentiation. So it's you want to pick on one single thing, and you're like, well, that doesn't make it multicellular. That's correct. All of these traits have to be present in the same organism to make it multicellular. There's not a single trait that makes something multicellular. So like, what is your definition of multicellular?
Article.
Again, no, that's what they say in different articles.
Well, because all the other article, that one article specifically, you say they branch off with one single cell and then start forming more clusters, and that's a multicell organism. But in all of the other articles, it says that they after they settle from centrifuge, they split right off into single cells. So no, that's not what all the articles say. You're showing one specific situation where you're saying they mutated into a multi-cell, but really it's just all of these single cells sticking together, going back to the,
Well,
big family analogy.
that's one part of being multicellular is your cells stick together. What is your definition of multicellular?
So a multi-cell organism is one organism that has multiple cells that are functioning different parts of the body. However,
Yeah, that's what we have.
That's okay, but
So
that's not what we have because they.
Do you accept that the Volvox carteri is a multicellular algae?
Yeah, so the only thing I know about the Volvox is when they compare the genomics between the Volvox and the Chlamydomonas. I haven't actually looked that deep into it, but yeah, it does appear that that is a multi-cell algae.
And that's because they have a soma, which is the body of non-reproductive cells held together by an extracellular matrix, and they have reproductive cells that have a different function. So the Volvox has its reproductive cells inside of like this bubble of soma.
Yep. Mm-hmm.
And the Reinhardtii does not have that bubble, but they still have cell differentiation. So really the only difference is the shape of their multicellularity.
No, not at all, because they're not the same organism. They're still single cells that are just sticking together. That's what you said. There's single cells that are sticking together.
The Volvox is made up of single cells that are sticking together.
No, they're inside of a body. They have a body. The soma, I believe you called it, that's the body. Just like our skin is the body that holds all of our cells or all of our organs inside of it.
The body doesn't have to look that nice. It can just be kind of mush. Like that's what a sponge is.
Right. I don't know too much about sponges, so I can't say too much about that.
Well, they're multicellular.
What you're saying is that all of these single cells stuck together as if, 8 people decided to put a hula hoop around them, and now that's a multicellular organism. No, it's still 8 people. They're just stuck together inside of a hula hoop. And yeah, they decided to take on roles of which one's going to reproduce and which one's going to keep the others alive, but they're still single cells that are stuck together. They never formed into one cell.
So then by your analogy, then as long as you form a 3D sphere of humans and put some humans inside it, and you only let those humans inside reproduce, that is now like a multicellular or single multicellular organism?
No, you totally lost.
This analogy breaks down because you can say this about any multicellular organism. It's just a bunch of single cells stuck together.
Yes, but in multi-cell organisms, they have specific functions that you can see that are part of their their genomic structure. So, for instance, the liver can do one function it can do multiple functions, but its main function cannot be done by the heart. The heart's functions cannot be done by the lungs. However, with your situation of all of these single cell Chlamydomonas sticking together, they could all take on each other's job. It's just that they've chosen which ones will do what job until they can branch off and reproduce. That's the difference. A multi-cell organism has specific parts that can only do a specific function, and if that function fails, or if that one organism or that one cell inside of that organism fails, then the rest of them crash down. However, in your scenario, if one of those Chlamydomonas ends up failing in its reproduction, it has other ones that can take on that role. So that's what I'm saying. There's
not
still
necessarily. So again, it is
1
on
minute,
the spectrum.
guys.
So you can see the difference because the reproductive cells have motile flagella, but there are organisms like planaria that are multicellular, and you can cut them in half, and their head will go on and survive.
You could.
Cells will revert back to stem cells to create whatever cells they need. So all of their cells can revert to stem and fulfill any function.
Okay, let's give you the final word in the discussion since Snake started. And then Snake, whatever you want to respond to, save it for your closing statement. You both get 5 minutes, and then we'll get into some audience questions. So Jamie, go ahead. Quick final word for the discussion, and then we're going into closings, gentlemen.
I mean, I think we've kind of already covered everything. We're just trying to get more details here and there, so we can go ahead and go to closings. That's fine.
Okay, Jamie, Snake, I appreciate the very engaging and technical discussion. We've hit over the 50 minutes. So time has flown. We got some excellent questions from the audience, and I appreciate you both making it easy to moderate, keeping it professional and civil. So with that, Snake, we're going to give you your 5-minute closing. So whenever you're ready, let me know, and I'll start the timer.
All right. 5 minutes. Yeah. So, I mean, I feel like I'll be repeating myself a lot, but so we seem to agree that on the basic components of what makes something multicellular, it's just that we don't agree on that it is multicellular. So I gave my definition. His definition seemed to fit mine as well, but he just doesn't agree that these traits are qualify as multicellular criteria. So I think it's this same paper we're talking about. No, if it's a different paper about Volvocine algae that talks about science is aware that there are intermediate degrees of individuality. So this would be pretty much the simplest form of individuality. So they do have cell differentiation, and I did actually add something to my list, which I didn't originally have in my opening, which is that they're all daughter cells of the same parent. They're not unrelated individuals aggregating. But back to the point is there are intermediate degrees of individuality, and that's shown in the various transitional forms of algae. There are actually, I think I'm going to share my screen for this one. So here next to the Volvox, which Jamie said was multicellular, which has meets all the same criteria as the Reinhardtii that we're talking about in this experiment. But next to it is the Pleiodorina. Probably butchering half of these names, but we're trying. The dark green is indicating germline cells. So these germline cells are actually on the surface of the bubble, which is gonidia, I think, is the proper term for that. Whereas Volvox, they're inside of the bubble. On Pleiodorina, they're on the outside. And in the Reinhardtii example, they can be inside the main body or on the surface. I don't think it matters. But there's a whole spectrum of differences in individuality. So all the way down to the gonium, we could actually, I don't think that looks like just a colonial cluster, non-multicellular. But if they do have cell differentiation, then that would qualify as simple multicellular. So these last four, or at least these last two, are definitely multicellular because they have cell differentiation and they have a body. And you can have that for single cell colonies, but that's because they're not related. So if they're all related from the same mother cell, if they're all daughter cells of the same cell, then that is a multicellular body right there. And this isn't the shape that we see in the experiment that we've been talking about mostly, but it has all of the same criteria. And the shape of the body does not matter for multicellularity. It only matters that you have, I added in here, same daughter cells. Limited cell number is not actually a requirement, but it's definitely distinct for multicellular. They're bound together by an extracellular matrix. They do not revert back to single cellular when put into conditions where their ancestors revert to single cellular. And they have cell differentiation, which have two different types of differentiation, germline versus somatic, and motile versus non-motile, respectively. So that is that's been observed in the lab. That's what we've seen from the experiment. I believe that I've met all of my criteria. I don't believe there's anything else that I really wanted to get to. And I think I'm out of time. So there you go.
There we go. That's 5 minutes. Snake was right. Thank you for the 5-minute closing statement. Jamie, we are now going to hand it to you. You also have your 5-minute concluding statement. So whenever you're ready, the floor is yours.
Yeah. So I'll keep it short, but I just want to say I had a blast doing this debate with you, Taylor. In fact, I would actually like to maybe have some conversations over emails, get some more details from you. That way I have more things to research for another debate in the future. Again, you've been studying this a lot longer than me. So of course there's going to be some things that, you have to explain to me, but I feel like it's fairly obvious that you're not really showing a multicell organism. You're just showing single cells that have stuck together to survive, and then they branch off, make single cells again. One thing I wanted to point out for the crowd was he said that cell differentiation between different daughter cells is still a single cell, but not if it's from the same daughter cell. That honestly doesn't prove anything to me, because that would be like saying that, this Chlamydomonas reproduced, and if those cells that are now single cell decided to stick together, well, they're a multi-cell now. That doesn't prove anything to me, because they're still single cells. If they don't stick together and they branch off and go and have their own babies and live their own lives, they're single cell. But because they decided to stick together, that makes them multi-cell. And I don't agree with that at all. Another thing that he mentioned was he compared a multi-cell to another multi-cell, and then he said, hey, look at these graphs that look similar. Again, that's like saying, bears and dogs are the same animal because they have 4 legs. Okay. I just don't accept that kind of evidence. So I'm sorry. But I thought it was interesting that he shows this multicell organism that has a wall. Each part of this multicell organism, the Volvox and the Pleiodorina, they have their own wall, structured wall. Each one of those cells has its own specific function, and it does that function specifically. If anything else tries to do that function, it will fail at that function. And if it fails at that function, then the whole cell or the whole organism fails. But then you look at a Chlamydomonas that sticks together, and you say that it has limited cells. Okay, well, that's great, but it doesn't always have the same limited cells. Just like a human will always have a lungs, a heart, 2 kidneys, 2 liver. I'm sorry, not 2 kidneys, liver. It'll always have those. But what you're saying is it has all of these different. It has all these cells that are sticking together, but it doesn't always have the same ones. They don't always serve the same function. And I mean, all I've seen so far is that they're just sticking together, basically. So I'm sorry. We'll have to talk more in emails or whatever whatever you feel comfortable with, so that I can get some more information on this, because I feel like you haven't really provided enough evidence that shows that it jumps the line from unicellular to multicellular. Yeah, at the beginning of the debate, I was not aware of all the terms that go into this. But as I dug deeper into your definitions, you really just showed me that it's single cells that stuck together. Whether or not they come from the same daughter cell or not doesn't mean anything to me, because it's just like the analogy I used with the Hispanic family. If 15 brothers and sisters decide to live in the same house, they're still 15 different people. And you're saying that the only difference between a single cell and a multicell is that the daughter cells are different. They're from different daughter cells. That's the only difference that makes the line of unicellular to multicellular. So I'm not fully convinced on your information that you provided, and I would love to talk to you more about this outside of the debate. So I'm gonna go ahead and wrap it up there. I will go back and watch all this, grab all the details that I may have missed, because I know I missed a lot, and I'll put together some more information for another presentation.
All right, Jamie, thank you so much for that concluding statement. Gentlemen, I got to say good job to the both of you for tackling and taking on this technical and important topic. So I always appreciate these hyper-specific debates where we can basically dig as deep as possible and be as comprehensive as possible. So you guys are... have worked hard and endured for 2 hours. So I'll give you a 30-second break here as I give some shout outs over some upcoming debates. We'll start specifically with the evolution topic. So the Evolution Debate Challenge series does continue with Wade the Wizard and Dr. Dino. So the saga continues. Evolution on Trial. We are going to be wrapping up the month with this debate. We've also got a round 2 between Ryan Adler and Ken Hovind. Evolution is on trial. Myself and Grayson will be having a round 2 on ancestry, the great ancestry debate. Does genetics support universal or common ancestry? If you want to see round 1 of that debate, which I highly enjoyed, check the description box of that video. We've also got the round 2 between Dr. Dino and Grayson. That'll be for next month as we do our best to appropriately space out these debates, as we are hosting several a week. So next month, the Great Age of the Earth debate, 2 titans in the creation versus evolution debate world, T-Rock, Mark Reed, and they will be debating the age of the earth. So a couple theology / soteriology related debates here to announce. So we've got Charles Jennings from the Layman Seminary. He'll be taking on Turretin fans. So this is definitely going to be a more scholarly debate. Both Charles and Turretin fan are well educated in this specific topic. The topic of soteriology, specifically lordship salvation versus free grace theology. And we've also got there it is, a debate on theistic determinism. So we've hosted over 240 debates on this channel on all sorts of topics, but I believe this is the first time we'll be doing a formal debate on this specific topic. Dan Chapa, he is a Molinist. CJ Cox holds to theistic determinism. And so we'll be debating the topic, is theistic determinism biblical? And we've got a fun one here coming up December 1st. I am pumped. Definitely a controversial one in the world of Christian theology, eschatology, and soteriology. Who is true Israel? We'll be discussing dispensationalism, replacement theology, the church, and spiritual Israel. David Preston and Pastor Matt First both know a ton on this topic. Pastor Matt First has written a book on this topic specifically. David Preston has debated this topic numerous times. So this will be definitely one to remember. And with that being said, break time is over. Jamie and Snake. So we are going to, we're going to jump right into the questions, gentlemen. So how we do it on this channel is whoever the question is for, we'll make sure that they get the last word. So Jamie, say the question is for you. You get to answer. Snake can add his thoughts. If you say something that he wants to object to, he can do so. And then Jamie, you would get the last word, and vice versa. So some of these questions are more so comments or just objections to what was said during the debate. And then we'll give you guys the opportunity to engage those. Okay, so first question that came in all the way near the beginning of, I believe, during the rebuttals. Dave H., thank you so much for your question. Question for the creation side. So that would be you, Jamie. Dave is asking, if everything is designed, why do we need to reproduce, as giving birth can and does kill the host? Jamie, just make sure to unmute yourself and you're good to go.
So I'm not exactly sure how he's trying to prove evolution with that. But I mean, you could turn that question around and say, if everything evolved, why did it evolve the ability to reproduce? Why didn't it just evolve the ability to live forever so that it doesn't have to compete for natural resources? I mean, I feel like this is kind of a gun with a hollow point bullet that has no gunpowder in it. He's shooting nothing. As far as why do we reproduce? Well, that's the way that it was designed. We had 2 humans that started everything. they reproduced with perfect genes until we got to a point where God put his judgment on the earth, and that's when the great flood came. So from there, they had to reproduce again. I mean, it's just a natural part of life. If you don't reproduce, then you will eventually die. Now, I imagine that we wouldn't have to reproduce if Adam and Eve never ate the apple and they just decided to live forever in a perfect creation. But since they ate that apple because of Snake, who was not right, now we have to reproduce to keep the civilization alive. So yeah, that's all I'm going to say on that.
All right, I appreciate the response there, Jamie, and thanks for the question, Dave. Snake, over to you. Floor is yours.
Well, I'll got a couple notes. I don't think that's much of a point against creationism, but I will say living forever is not good for the population at all. So I think there's a reason that is kind of selected against, because the one who lives forever will compete with others for resources, and kind of the longer you live, the more it's like the Pareto distribution. If you're successful, you're going to be more and more and more successful over time. So those who live forever are going to outcompete the younger ones for resources. So also, if one individual keeps reproducing or stays around and keeps reproducing, the population is not going to evolve very fast either. So evolution would select against that, because not evolving also means that the environment can change. Your population might die out faster. But so to the spirit of the question, I would say better examples would be like the hornets that need to lay eggs in their hosts, or it's like a caterpillar that infects the eyeballs of snails and needs to be eaten by a bird to reproduce, and like hermit crabs, their entire morphology is based around using shells of dead organisms. So their designs are all based on death needing to happen, which was not the conditions in which life was originally designed. There was no death, apparently. So I think these are better objections to that kind of thing. Doesn't make sense how they were designed type of arguments.
Okay, thank you so much there, Snake. Jamie, question was for you. We'll give you the last word.
So he said a couple things there that I feel would be pretty interesting to cover. First, he said living forever is not good for the population. Again, who cares if it's good for the population if it's the first organism that was ever created or that ever evolved? Let's say that, one Chlamydomonas evolved into a sea sponge. If that sea sponge can have all the resources to itself, who cares about populations? Another thing was he says that the reason that they evolve is because they get better over time. However, that's not what we see at all. We see that thousands of years ago, humans had a much better understanding of astronomy and astrology, and now we have no understanding of it. Same thing with humans lived longer. They were healthier. They were taller. And now they are weaker. They get sicker. There's more diseases. So over time, we actually get worse. So that goes against your whole argument of that point. The other thing was the hermit crabs need dead shells. I thought that was interesting. I've never heard that argument before. But I don't know if you know this or not, but there's plenty of shells that don't come from humans or from other dead organisms. There's shells that just exist. But what's more interesting about that is the system that is required. That would be considered a symbiotic relationship, if I'm getting the term correctly, where the hermit crab can only survive if it has something to go. And if it grows and it needs a shell, it needs to find a shell to survive. So therefore, that's I'm sorry, it's not a symbiotic. It's I'm forgetting my terms. Mutualistic relationship, whatever the term is. But again, what would happen if the hermit crabs evolved first and it didn't have something that needed a shell, or it didn't have something that gave a shell away? So you see, there's plenty of mutualistic relationships in nature that just don't make sense with evolution. Another good one would be bees and flowers. How does the flower survive without the bee? And how does the bee, what's the purpose of the bee if there's no flowers? See, there's plenty of situations where it wouldn't make sense in light of evolution, but it makes sense with design. So that's it.
Okay,
Well,
thank you.
If I could add real quick, and you'll get to respond. So the thing with the immortality is, like, if something could evolve that, it would have to go through a lot of previous populations first. And so evolution would already be kicked in, and population genetics would already be important. And the other thing is, like, what happens if you have a population of one immortal thing, it can still die if it runs out of food, or it's like elvish immortality. Like, they can be killed, but they'll if they don't get killed, they'll live forever. So that's where, like, altruistic traits come in, like, which is happening on a micro scale in these multicellular experiments. Is some cells take a hit in fitness so that they benefit the whole unit, which can be a body, it can be a population, etc.
So yeah, I'll entertain the hypothetical here. Yeah, if it were to run out of food or something like that, and it dies, then yeah, that population or that one is gone, I guess, because it wouldn't be a population at that point. But I mean, why wouldn't it evolve? Okay, we hear the legends of the phoenix that when it dies, it catches on fires, burns to ashes, and then it resurrects itself from the ashes. Why didn't evolution
There
do
are
that?
jellyfish. Sorry, there are jellyfish that kind of do that.
Yeah. Okay. So see, there you go. Like, why didn't all the animals just do that instead of, reproduce and then compete for resources? It's a fun hypothetical to entertain, but again, it's pretty obvious that reproduction is essential for the creation design as well as for evolution design. So I guess we can both agree on that.
Okay, guys, thanks for keeping that question fun. Some good engagement there. And all right, we'll move on. This one is a super chat, comes in from Bubblegum Gun. Snake, your best buddy here, and he's coming at you with more so a comment, and therefore we'll give you the chance to respond. So Bubblegum says the paper snake says proto multi-cell, not multi-cell.
Some of them do. Let me check. Nope, it doesn't. It says something about protocol, but that's as close as the main paper we were discussing gets.
Okay, thank you, Snake. Jamie, anything you want to add?
Nah, I'd have to look into that, so I'm not going to add anything for now.
No worries, no worries. Okay, so now we got a comment or an argument for you, Jamie. This one comes in from Grayson Audette. Thank you so much. Grayson is asking, Jamie, the difference is, in this case, the single cells of Chlamydomonas are no longer competing with each other or reproducing independently. Not single cell, it's multi. What are your thoughts on that, Jamie?
Well, he says that they're not reproducing independently, but that's exactly what happens. They branch off. A single cell jumps out of this cluster, and then it reproduces independently to create another cluster, according to what the paper is saying. So that's not exactly true. As far as no longer competing with each other, I mean, the algae don't really have to compete with each other. They just kind of live and replicate. So what are they competing for?
Okay, appreciate the response there, Jamie. Snake, yeah, you get the last, or no, well, it's for you, Jamie, but Snake, we'll give you the opportunity to respond. Go ahead.
So yeah, that's a good point. I don't think I mentioned is they aren't competing with each other anymore. So that's part of the altruistic trait is, they take a hit in fitness because they can't reproduce themselves anymore, but now they're taking care of each other. And that's an important part of multicellularity. And individuals in a population can certainly do that. Like, if you're related to your brothers and sisters, you can help each other. But you're not bound up in the same body, and your brothers and sisters aren't like, you're designated to reproduce, but you're not. That's since some cells are designated for reproduction and others are not, that would be the difference there.
Okay, thank you, Snake. Jamie, you get the last word. Go ahead.
I know. I think I have said all I need to say on that one. So
all right. Appreciate it. This one, next one comes in from Hive Science. Thank you so much for your question. And Hive Science is asking, Jamie, does Jamie realize that he keeps describing zooid reproduction and claiming that this form of organismal propagations is not observed.
Yeah, I'm not exactly sure what he's asking there, so I guess I'll just skip over this question.
No worries. No worries. Snake, anything you want to add at all?
Yeah, I mean, I think that you were confusing some of those phases with what was actually observed, which, that they wrote on why this observation was novel. And yeah, good to see you, Bree. How's it going?
And it looks like Zach Hancock, appreciate it. So maybe it's pronounced zooid. Zooid. There we go.
Yeah.
So, okay. Next question comes in from Tanya Brown. Tanya,
Well,
thank
hold
you
on.
so much.
I do want to add one thing to that last question.
Yeah, Jamie, go ahead. You get the last word.
So he said that I wasn't understanding what was novel about it, and that's literally what I was asking him for almost a whole hour. Is what's the difference between that and all the other reproductive processes that have been observed? And all he could say was, well, it's obligate. Okay, well, that doesn't explain the difference between it. That doesn't show the line that jumps from unicellular to multicellular. So if that's what they were asking in the question, then I mean, yeah, he never showed what makes it different to where now it's a multicell organism. He said it's obligate. He said the cells stick together, and then he just kept circling back to those two answers.
Well, the zooid specifically shares a cell wall, so that's one other difference.
But these ones don't. They all have their own cell wall.
In the experiment, correct.
Right. So
that's another difference. That's a difference between zooid and the experimental phase.
Right, but that's what this whole argument you were saying, oh, well, they have their own cell walls, but they're inside of their matrix that makes them a body, but they don't have a structured body. They don't have specific functions, and then when they go to reproduce, it makes that exact same cluster. It's just more clusters of single cells. Again, that's the matrix. That's what I was saying with the body. When I reproduce with a female, the baby that comes out is going to have two sets of eyes, or it's going to have two eyes. It's going to have two lungs. It's going to have a liver, a kidney, organ. It's going to have all these same components every single time. That's not what you showed in those articles. You said, yeah, they branch off and they create another cluster of single cells that stick together, but they don't have the exact same cells with the exact same function. So therefore, it's not multicellular.
Well, they did have two different types of cells every time. The difference was number of cells, which varies in all multicellular organisms, or at least most that I know of. And yeah, I forget what your other point was, but that is the extracellular matrix. That is a protein that's binding all the cells together in one body.
Right, but it's not a body though. It's just the matrix, and that's what you said multiple times. You were saying, yeah, it's not a body, it's a matrix. So that when I pulled up aggregation, you said, oh, that's not a body, that's a matrix. Like, okay, well, still same thing. Like
the cells
and
the matrix that as body.
Yeah, but you want to but when I use the example of the aggregation from the chart, you said, oh, that's not a body, that's a matrix. So therefore, that somehow disproved that it's not the same thing. But then you want to turn around. Right, but then you want to turn around and use that exact same argument to prove that it is multicellular and it is one unit. So, yeah, you're just circling around on the same term.
But there's differences on each comparison.
Okay. Okay. People can see through that, but okay.
All right, guys, keeping this fun. I appreciate it. So next question comes in from Tanya Brown. Tanya, thanks for the question. And the question's for Snake. If all of this is from pre-existing DNA, don't these experiments demonstrate epigenetics and selection rather than evolution?
No, because epigenetics is not necessarily, or it is not a actual genetic change. That would be, what is it? I forget the word for it, but it basically there's things that stick to the DNA that upregulate or downregulate their expression, and that would be epigenetic. And so rather than evolution. And the existing DNA claims is that is how evolution works. There's nothing that just suddenly pops up with a new gene, that the genetic material that the parents didn't have at least part of. So all of evolution is a modification of something else. The only exception would be like viral insertions and bacterial transfer of plasmids. That's horizontal gene transfer. But again, that always comes from a pre-existing line of code. It doesn't have to come from the parent, but it's always pre-existing. The difference is that when you change the code, sometimes it has a different function. Sometimes that function is really bad. Sometimes that function is good. And it largely depends on context.
Okay, thank you, Snake, for the response. Jamie, over to you. The floor is yours.
Yeah, I believe I asked him this a couple times during the debate. I mean, it really just seemed like artificial selection. And I know I asked him, where's the new genetic information? He said that whenever evolution creates a new organism, there is no new genetic information. But that's not true, because we can clearly see that, for example, a bird has genetic information that gives it a wing, but a fish has genetic information that gives it a gill. Those are two different pieces of genetic information. So how did that happen? See, you want to say that, the DNA will just slightly modify, and then over billions of years, well, boom, there's your fairy tale over billions of years, because there is new gene expression. So that's what I'm saying. How is this a multi-cell when it has all the exact same DNA sequences? That's what I think we kind of started touching that when I mentioned, or when he mentioned the Reg A like gene. And then I pressed him on that. Was it before the experiment? And he said, yes. Is it after the experiment? Yes. Okay. So there was nothing new there. In order for it to be a new organism, it has to have new information. And he didn't show any new information. So yeah, it just seems like natural or artificial selection.
So it is artificial selection, which doesn't mean it's not evolution. So there is something new there, because the gene is not exactly the same. It's not exactly the same as the wild type either. They undergo mutations, which changes the way it functions, or changes the way how much is produced, and how much is there actually can change the information. If you have or when it turns on, even you can have the exact same gene and have it respond to different stimuli, and you can have different traits. So in this case, the gene that terminates cell division is being regulated to turn off in most of the, or to turn on, which turns off cell division in most of the cells, creating a non-reproductive cell body. And then some cells are designated to not have that gene turn on, and those reproduce. So you're getting, and the end result of this is a body that has cell differentiation. So this is a new trait from existing information just being regulated differently. So this is new information. And for evolution in general, all we have to explain is the differences between transitional forms. So can one organism, have its bones fuse and get longer bones? Can it get a shorter tail? These things are all explained by microevolution. And if you put enough of them together, then it, of course, gives us larger changes, more noticeable changes. But so since we know each individual step is possible, and we know multiple steps can combine in the same population, and these steps have been predicted before they were found by evolution, this is a scientific confirmation of evolution.
Okay. Did you guys want to go back and forth one more time on that, or I wasn't sure, Jamie, if you're going to respond?
Yeah, he said a lot in that, but the one thing I just kind of want to point out is he says, "Oh well, if it's being used to support creation, then no, that's not right. But if it's being used to support evolution, then yes, that's correct." I can't remember exactly what it was he said, but he went back and forth on it. And when I tried to use it for evidence for not being evolution, he said, oh no, that's not right. But then he goes and turns it around and uses it to be evidence even after he said it's not right. So I mean, the people can see that, you know, you're seesawing between definitions. You take the data, and if it fits what you can explain, if it fits your ideology, then you'll accept it. But if it doesn't fit your ideology, then you'll reject it, even if it's the exact same information you just accepted. So I thought that was kind of funny.
Well, see, it's the method and not the conclusion. So you take the data and see if it fits with your creationist story. Evolution, on the other hand, can actually predict the data, which is the method supporting the conclusion versus just you're trying to fit the data into your conclusion. So that's the major difference.
That's not true. That's not 100% true, because there's plenty of creation scientists that are making very successful predictions today, and their peer-reviewed papers are getting shot down by the mainstream media. The perfect example would be Nathaniel Jensen and all of his work that he's doing with Retraced. He's showing us the migration of humans from the original source at Babel, and yet you reject it because it doesn't fit your theory. That's because you have a presupposed idea of what you want to believe. Another good example that's not a creationist is Graham Hancock. Graham Hancock doesn't believe in Christianity. He doesn't believe in any religion that I know of, but he believes that there was a great flood. And he shows plenty of evidence in his documentary, Ancient Apocalypse, and yet the evolution community shoots it down because they have a presupposed conclusion that they have to enforce for whatever reason. I know what the reason is, but I'm not going to get into that because that goes into conspiracies. However, we can see that over and over again, whether it's a creationist or whether it's a secular scientist that just has a different theory of the history of humanity. If it doesn't fit evolution, the peer-reviewed editors shut it down.
So with Jensen, his methods are flawed, and
of course they are, because they're creationist, right?
Well, I think it's more the other way around. He's a creationist, therefore his methods are flawed.
Just exactly what I said.
More along those lines, because no, his methods are flawed. No one says you're a creationist, therefore it's wrong.
That's exactly what you said.
If you want a more detailed breakdown of why his methods are wrong, you can look at Dan Cardinale's channel, Creation Myths.
Dodgeball Dan. Okay, yeah, I trust everything he says.
The guy who actually confronted Jensen face to face, who got Jensen to admit that his methods are not the correct methods. Yeah. As for Graham Hancock, I'm kind of a fan of Graham Hancock. I think his stuff is interesting, but he's not a scientist. Who's the guy who's with him?
I don't
think he's a scientist either. But they propose more of a series of great floods, not a single one. But challenges to evolution are published all the time. You just have to read enough literature.
Yeah, if you dig deep enough, maybe about 100 years ago, you'll find one article. But what's interesting about that is
recent.
What's interesting about that is you said I actually lost my train of thought on that. It was something about Graham Hancock. But yeah, I'll just go ahead and leave it at that. That's fine. You can have your last word.
Go ahead, Snake. Quick final word.
Go ahead. Let's go to the next question.
Okay, guys.
I need to start writing things down. I'm not. This is my first debate, so I forget to write things down because I think I'm going to remember them. So yeah, I need to write things down more.
Good old pen and paper.
Yep.
Definitely comes in handy in a debate. Well, guys, no, you kept it fun. It was an excellent debate. Let's wrap it up with one more question, and then that way we'll wrap up the debate around the 2.5 hour mark. So I do want to thank you both for the time you've given to us tonight. This one comes in from Shannon Collins. Shannon has stated that there's a typo in here, so I'll fix it. So Shannon's asking, question for Snake, was there ever an event where it did not shoot? So instead of did, should be did not, I believe, shoot of a single cell to multi-cell. Or does that question make sense to you, Snake?
Yeah, I think so. So in the beginning of the experiment, yes, some of the colonies were still wild type. They selected the ones that were, mutant type, multicellular type, and they bred those together. And some of them were still wild type because it's a population in transition, the entire population had not yet rolled over to all having the same mutation. So yeah, they had that all throughout the experiment. They eventually got to a point where it was all multicellular type and no wild type.
Thank you, Snake. Jamie, if there's anything you'd like to add.
Yeah, I mean, if I was going to add anything to it, I would just go back to the point that because the single cells are sticking together doesn't make them multi-cell. So the question in itself is invalid. But yeah, if you want to keep calling it multi-cell, go ahead. I'm going to go after this debate. I'm going to go glue myself to my brother, and we're going to become a new organism.
All right.
Well, I wish you luck, but to clarify, that, so it wasn't that they only had the multicellular type because they just selected them out and that's all they kept. Is each time they did this, each round of doing this, they got more and more multicell type and less wild type being born out of this population. So they were selecting based on, the artificial selection that they were trying to make more of this multicellular type, but eventually they weren't giving birth at all to any other wild type because that had been bred out of the population. It wasn't that, they just took it out and they're like, look, in this box, I only have multicellular. No, those multicellular eventually they only produced multicellular and no single cellular anymore.
Well, I mean, the more generations you get in a lab, of course, you're going to have less and less wild cells. Like that's because they're selecting out the wild cells. So, I mean, that's not a breakthrough. That's just them selecting out wild cells until they have a cluster that they can call something different.
Yeah, because they're genetically different. So they produce genetically different children. And thus, you got a different organism.
But it's still single cells sticking together. Like I said, I'm going to glue myself to my brother. Same thing.
Okay,
And,
you guys.
And that's where we disagree.
Yeah, that's where we disagree.
Oh, who's going to get the last word on this one?
Yeah, that's him. He can have his last word.
There you go. That's all I got.
There we go. So we kept to the format. So Jamie, Taylor, thanks so much for the debate. Excellent debate. Please, to the audience, share this around. We strongly believe in critical thinking skills, which is why we like to host these kinds of debates. These are important topics, technical topics, but they need to be engaged. They need to be discussed. So again, Jamie and Taylor, thanks so much. Let's have some quick final words and final thoughts. Jamie, why don't we start with you? Thanks again for doing this. And final words, final thoughts.
Yeah, I would just like to say thank you for taking the time, Taylor. I'm not anybody special, so I really appreciate you taking the time to do this debate. And also, thank you for adding a lot of new things for me to research, because I noticed that the few papers that I could find, they kind of just repeated the same thing, and you brought a lot of new stuff to the table. So that gives me something to research, and that'll give me some entertainment for the future. But I would like to extend out an email or something and, get some more information from you, maybe do a back and forth through emails, and, maybe we can kind of clarify and find some kind of common ground, because you're a smart guy. The only thing is you've been convinced of something that I don't believe is true, and I don't believe there's evidence to prove that it is true. So I will say that I don't think you're dumb. I don't think you're ignorant on the topic at all. I just think that you have presupposed conclusions that you want to be true, and that's where we see two different endings. So thank you for the time, and I really appreciate you taking the time to do this, man.
Mm-hmm.
I appreciate the final words there, Jamie. And yeah, Snake, go ahead. Final words, final thoughts.
Yeah, I mean, I think the same, that you probably want your conclusions to be true as well. I had a good time. It was fun. You're a good guy, and thanks for the debate. And hopefully, I wasn't trying to give you a hard time too much. But it's just, scientific topics can get over people's heads sometimes. So and absolutely, I would go for more rounds on this channel, any channel. and I'll shoot you an email because I think I have your email from the challenge that you issued. So yeah.
Yep.
Or Donnie, feel free to give it out. It's I mean, anyone can email me. It's at the snake was right at Gmail. So
Okay, appreciate it, Snake. Again, to the audience, I do have both of our guests, our debaters for tonight. I've got their links in the description box. If you like what you heard and seen from them, please do check out their channels. And a round two, I'm sure people will love to see in the future. So we'll definitely discuss that. Anyways, Jamie, Taylor, thank you so much for doing this. To the audience, thanks for tuning in. And thanks for all your questions. Stand for Truth is out.